Oh, Katie, I saw you. Okay, Katie's here. We'll just wait for the others. Beacon, got you. Justin. Yes, Justin, you have to click on the link that is on Moodle or on the Learning Hub to join the Hangout. It will take you to a YouTube link, a YouTube page, and you should be able to see the Hangout. I can't see who is in the Hangout. That is why we're using poll everywhere. So once you've joined the Hangout and you sign in, then I know that you're there. I'm unable to see who's in the Hangout because I'm using Hangout on air. I'm using Hangout on air, so it doesn't allow me to see who is in the Hangout, but it broadcasts it. Are you able to see me, Justin? Okay, great. Jasmine, I see you. And Corel is here. Oh, Learning Hub is slow. Sorry. Okay, so let's see. 1, 2, 3, 4, 5. Only 5 individuals. 6. Caesar, I see you. I think we should have about 10 today. Or 9 or 10. One, two, three. Okay, I'll give it 2 more minutes and then we're gonna get started with whoever is here. Yes, last lab is today. The lab today is on the nervous system, which is what we will begin to cover today. Taylor, I see you. Great. Yes, there is an entrance quiz. Okay. So you're saying that LH, who is LH? I'm not sure who's N. Oh, you're saying the Wi-Fi is bad in Lamson Hall, I guess. And Gadiel, okay, I see you here. Okay, so we're good to go. I think that's it. Well, we have to go. It's 11:35. Let's get started. We're talking today about the nervous system. And I am going to put up the slides, and I would like you to let me know if you're able to see the slides. Oh, Lamar. Okay, Lamar is here. Very good. Okay, it's slow. I don't know why. I've heard other complaints too. So here we go. The chapter for quiz today is on the nervous system. Okay, let me know if you're able to see the slide. So I'm sharing the screen. Let me know if you're able to see the slide, my screen. Okay, great. So let's go. Let's proceed with the lecture. All right, so we're looking at, so far, we've looked at several organ systems. So we're close to the end of the semester, as you know. So we looked at the digestive, the lymphatic, cardiovascular, respiratory, and urinary systems. And those all play a significant role in homeostasis. The skeletal and muscular system, although they do play a role in homeostasis, their role mainly is in support and movement of the human body. We will end the semester by looking at two systems that are important or necessary for coordinating and regulating all the other systems that we've covered so far. So those are the nervous system and the endocrine system. So I would like to cover the nervous system this week. Next week we'll do the endocrine system. No, next week is Thanksgiving. So after Thanksgiving, we will do the endocrine system. And recall, we have our test on next Monday. Okay, so let's bear that in mind. Okay, let me see if there are any questions so far. Okay, if you can't see the slides, I have posted the slides on the Learning Hub. So you could open the slide on the Learning Hub and follow along that way also. Okay? So I will continue. So we're looking at the nervous system, and it's organized into two main components. We have our central nervous system, which is comprised of the brain and spinal cord, and our peripheral nervous system, which is everything outside of the central, and that is comprised of nerves and ganglia. Nerves and ganglia. And just looking at this image here, you'll see how it's separated. So our brain and spinal cord, that's our central nervous system. And anything outside of that, these are our nerves that are projecting off the spinal cord. These are nerves projecting off the spinal cord. These are that's part of our peripheral nervous system. Peripheral nervous system. You would also see you can't see ganglia here, but ganglia are just clusters of cell bodies that are part of these. Well, actually make up the nerves. So the nerves and ganglia are the peripheral. The central are the brain and the spinal cord. Let's look at some of the cells of the nervous system. We covered this, we mentioned it briefly earlier in the semester when we looked at various types of tissues. We had looked at the 4 types of tissues and nervous tissue being one. Let's look at the cells that make up the nervous system. We have neurons, and you recall neurons have this very odd shape. They have, typically you have your cell, the nucleus that you would find in every other cell, but they have a cell body from which you have processes known as dendrites projecting off the cell body. So these are dendrites projecting off the cell body. And extending from the cell body, you have a very long process known as an axon. Okay, so that's a typical neuron. The cell body with dendrites projecting off of it, and we have a very long process known as an axon that projects off the cell body, that extends off the cell body. So we have axons and dendrites making up the neurons. And then you also have supporting cells that help to nourish and support the neurons, which include your oligodendrocytes. You also have astrocytes and microglia. So these are the cells that make up the nervous system. And here's a micrograph that shows you really nice, cool figure of neurons and microglia here. Okay, so these are all neurons. You can see dendrites projecting off the cell body, and here's an axon there. Okay, there are 3 types of neurons. 3 types of neurons. You have what are called sensory neurons. Sensory neurons are neurons that take information from the environment and send it to the brain and spinal cord. So here's an example of a sensory neuron. This is a sensory neuron, and you can see the sensory neuron is attached to a receptor in the skin. So sensory neurons take information from the environment, external or even internal environment, and sends it to the brain and spinal cord. So here we have a sensory neuron attached to the skin, and it connects to an interneuron. Interneurons are found in the central nervous system. So you'll find interneurons in the central nervous system. So this interneurons, they would receive information from the sensory neurons. They receive that information, and they will then interpret that and send it to a motor neuron. Motor neurons will take information or take impulses from the interneurons, which are in the central nervous system, and transfer them to an effector. An effector could be either a muscle or a gland. In this figure, you're seeing the motor neuron is attached to a muscle. Okay, so information comes in. Comes in via the sensory neuron. It's processed in the interneuron, and then signals are sent out via the motor neurons to an effector. The effector, again, could be the muscle, could be a muscle, skeletal muscle typically, or it could be a smooth muscle, any muscle actually. And then or it could be, an effector could also be a gland, okay? Causes a gland to secrete once it gets that signal from the motor neuron. Okay. All right, so I see Yasmin and Darrell are here. Okay, welcome. Okay, let's continue. Before I continue, let me ask, oh, never mind. I'll just continue. right, so who is this? This is Lou Gehrig, famous baseball player, okay? And he was famous for baseball. He was also famous for a disease known as Lou Gehrig's disease, or ALS. And let's see what happens. In ALS, motor neurons are degraded, they're atrophied. So here's a normal neuron, and it's attached to a muscle. But in ALS stands for amylo, I can't remember. I can't remember offhand, but it's a long name. But anyway, this disease causes motor neurons to atrophy, so they break down and they're not functioning well. And as a result, it causes the muscle to which they're attached to atrophy. Okay, so the muscles atrophy over time. So with individuals with ALS, they could receive information because their sensory neurons are working fine. So they could get information from their environment. They could see, they could smell, they could touch, because information is still, impulses are still coming in from the environment. Those impulses could travel to the interneurons, and that information could be interpreted in the brain and spinal cord. But then that impulse can't travel to the motor neuron because the motor neurons get broken down over time. And with that, and the muscles to which that motor neuron is attached to, or the gland to which that motor neuron is attached to, doesn't function over time. And so you can have muscle being atrophied. So individuals with ALS, after a while, they lose the ability to move. They lose the ability to walk. They cannot move. They end up in bed, and eventually they can die because the diaphragm. A lot of times for these individuals, the diaphragm also atrophies, and we know that the diaphragm plays a significant role in breathing. So if your diaphragm atrophies, then you eventually will stop breathing. So ALS it's very, very serious condition. Okay. Let's look also here. I want to show you a neuron. Interestingly, if you recall, let's go. Neurons have axons extending from them. So let me go back to the figure here of an axon. A neuron. Sorry. Okay, so here is a neuron, okay, with the dendrite projecting, and here is our axon, okay? So we're going to zoom in on that axon and see what it looks like close up. So here's an axon. Axons are surrounded by what are called myelin sheets. See that? Myelin sheets are special cells called Schwann cells that wrap around the axon. So you see here, there's a host of myelin sheets wrapped around, or Schwann cells which make up the myelin sheet, wrapping around the axon, the entire length of the axon. And I wanted to show that because I wanted to mention another degenerative or neurodegenerative disease known as MS, multiple sclerosis. And I'm sure you've heard of multiple sclerosis. You may actually even know someone with multiple sclerosis. In multiple sclerosis, what happens is that this myelin sheet is broken down. And the myelin sheet helps the nerve impulses to travel fast along the axon. But in individuals with multiple sclerosis, this myelin sheet is broken down, and as a result, the impulses travel very, very slowly through the axon, and that causes a degenerative disease. Okay? All right. So these neurons, although you can't see them, they look quite simple. They are actually very, very crucial. I want to show you here. It's another. That's actually a nerve cell. So you're seeing here the cell body. These are all dendrites projecting off the cell body. And here's the axon that's extending this way, right? Another look of the axon here. See, that's the axon. You can see the myelin sheets surrounding it there. Okay. So let's talk about the significance of this myelin sheet. It plays a very important role in the conduction of impulses along the axon. And I'll just show you some text here that talks about that. So the myelin sheets are formed by Schwann cells. These are cells that wrap around the axon, and they allow for what is known as saltatory conduction, the conduction of impulses from one node of Ranvier to the next. So what is this node of Ranvier? You see that gap between the myelin sheath here? So we have Schwann cells wrapped around here, some more Schwann cells there, some more Schwann cells there. The gap in between them is known as, is called the nodes of Ranvier. So when an impulse is traveling along the length of an axon, it actually, if the myelin sheets are present, the impulses will jump over the myelin sheet and just jump in between the nodes of Ranvier. And that allows for the conduction of impulses to be a lot quicker, because that process, that jumping from one node to the next, is called saltatory conduction. Let me see, I think I might have a slide that shows this a little better. Yeah, here we go. Saltatory conduction. So here we have, these are our myelin sheets formed by the Schwann cells, okay? And so when impulses are traveling down the axon, it moves from one node here to another. So it jumps from one to the next, jumps from one to the next. And that is known as saltatory conduction, jumping from one node to the next. And that allows the movement of impulses a lot quicker than if it was not present. Okay. I'm going to stop and ask if you have any questions. Oh, you're still not seeing, Katie? Okay. I posted the slides for today on the Learning Hub. You can access it that way. Amyotrophic lateral sclerosis, that is ALS. Very good, thank you, whoever put that up. The slides, you cannot follow along on the slides that I'm posting. There's a problem with Hangout. It causes the slides to freeze, or any video doesn't show as it should. So that is why I posted all slides and videos on the Learning Hub, so you could follow along with the slide that I posted, not with the lecture, not with my presentation. You have to open the slides that are on Moodle. There's a link on the Moodle that says lecture slides. That is the one you want to open up. Please let me know when you find that link that says lecture slides and you're able to open it. So I'm going to wait for your response on that. Okay, at this point, there is no cure for ALS. No cure for ALS. If you have it, you die a very slow and horrible death. Did anyone else find the slides? The link that says lecture slides. I need to hear from someone else. Katie, I know Katie found it. Is there anyone else who found the slides? Gadiel, you said the slides are not changing, but like I said, it's on the link that says lecture slides. That should change. Or you will be able to manipulate that, I mean. Okay, great. So let's continue. Okay, so let's look at how an impulse travels. How an impulse travels down. Okay, let's clear that. All right, very good. Everyone seems to have gotten it. Okay, so let's see, what is a nerve impulse? So these nerve impulses are actually electrical charges. Electrical charges. So here we're looking at a cross section of a membrane, of an axon. Right? So the axon is really a long tube, okay? And here's a cross section of that axon. And on the inside, when an axon is at rest, when there's no impulse traveling through it, when an axon is at rest, the inside of the axon is negatively charged and the outside is positively charged. Okay? Inside negative, outside positive. The reason being is that it says there are more positive sodium ions outside than inside and more positive potassium ions inside than outside. Here's the thing I want you to remember. Don't get too caught up as to where the sodium is or where the potassium is. What I want you to remember, key thing, is that at rest, when an axon is at rest, when there's no nerve impulse traveling through a neuron, the inside of that neuron is negatively charged and the outside is positively charged. And that is referred to as resting potential. Resting potential. No nerve, no impulses traveling through that nerve. Then, when stimulation occurs, the neuron moves to what is known as an action potential. An action potential takes place. During an action potential, the inside of the neuron becomes, or the inside of the axon becomes positive, and the outside becomes negative. So what happens is that the sodium channels open and they move, the sodium ions move, the potassium ions move, and that results in the inside of the axon becoming positive and the outside becoming negative. So there's a switch. So at resting potential, the inside is negative, outside is positive. Upon stimulation, when the nerve receives an impulse, that charge switches around where the inside becomes positive and the outside becomes negative. The inside actually becomes positively plus 40 is the charge on the inside. So that's known as an action potential. Action potential. Once the charge passes down the entire neuron, then the cell, I'm sorry, we go back, oops, see, okay. The neuron then goes on to what is known as repolarization, where the initial positive on the negative on the inside is reestablished. So again, at rest, which is also known as resting potential, the inside of the neuron is negative, the outside is positive. Upon receiving an impulse, the inside of the neuron then becomes negative and the outside is positive. And then once the impulse passes, the neuron undergoes what is known as repolarization, where the inside becomes negative again and the outside becomes positive again. So I have a video I posted that I think would be helpful for you to take a look at. It's called the nerve impulse. I want you to look at that. That shows you how an impulse, which is really an electrical current, how it travels down the entire length of the axon, of the dendrites and axon, and how the changes in voltage occurs between how the change from positive to negative occurs. Let me ask you a question here. Let me look at the poll. Have you guys seen the videos that I posted? You don't not whether you looked at them, but have you seen the links? I should ask. Have you seen the links for the videos I posted? They should be on the Learning Hub also. Please let me know. Okay, great. So after class, you could take a look at those videos because it will help to clarify some of the things that we're talking about today. Okay. All right. Okay, so we talked about how the impulse travels and then saltatory conduction occurs. Okay, so here we see an action potential at work. So you see the negative on the outside, positive on the inside, and then that moves, that impulse then jumps over the myelin sheets and move this way. Okay, right. And then here is repolarization occurring at the bottom. Okay. Here's another video you want you to pay attention to. It's called the voltage gated channels and the action potential. So you want to look at that. Okay. Okay. So now let's talk about once the impulse travels down the length of an axon, how does it then get from one axon to the next? How does impulse travel from one axon to the next? Well, first let's define something. Between the junction, I should say, between one axon and the adjacent dendrite of another neuron is known as a synapse. So the junction again between the axon of one neuron and the dendrite of another neuron is called a synapse. Okay, and a synapse could occur between two neurons, but a synapse could also occur between the axon of a neuron and, say, a muscle. That's a muscular synapse, a neuromuscular junction, for example. But we want to focus on the junction between two neurons. That's known as a synapse. So here's a closer look of a synapse. So here we have one neuron. Here's a neuron, okay? And attached to that, that's the cell body of the neuron. Here's the axon of the neuron, okay? And you're seeing a few dendrites of the neuron. But attached to the cell body of that neuron, you see that there's several axons there attached to the cell body. So information comes down from this axon, for example, and it connects here with the cell body or dendrite of an adjacent neuron. Okay, that junction here between the neuron, the cell body or the dendrite, it could be a dendrite here, and the adjacent neuron is known as a synapse. Okay, let's take another look. This is an electron micrograph showing you synapse. Okay, so here the axon terminals, which just mean the ends of the axon, and you can see that they're connected to the neuron cell body. Okay, this is a drawing showing you the synapse. So here, this is the axon bulb. Okay, and so imagine that the impulse traveled all the way down to the axon bulb here. Okay, and this is the synapse, the junction between the axon and the dendrite or cell body of the adjacent neuron. And you can see what is known as a little space here. That's called the synaptic cleft. That's the synaptic cleft. The incoming neuron is known as the presynaptic neuron. That's the incoming. This is the axon of the first neuron or the previous neuron. This is known as the presynaptic neuron. Okay, and then your synaptic cleft is here, and then you have your postsynaptic neuron, right? Okay. All right. So how does impulses travel? How does that action potential travel from one neuron to the next? So imagine we had an action potential traveling along the length of this axon. So meaning that the inside changed from the -70 to now +40, and that travels all the way along the length of the axon. Well, what happens is that it causes, that action potential causes what are called neurotransmitters, which are found in tiny vesicles known as synaptic vesicles, to be released. So it causes these neurotransmitters that are in the synaptic vesicles to get to the synaptic cleft and release their content. And you're seeing here, synaptic vesicle containing neurotransmitters. They get to the synaptic cleft and release their contents into the synaptic cleft. These neurotransmitters will then bind to the adjacent, that's the postsynaptic neuron, and those neurotransmitters, would initiate an action potential in the adjacent neuron. So what's doing the actual initiation? What's doing the actual transfer, I should say, of this impulse from one neuron to the next? It's the neurotransmitters. Think about the word neurotransmitters, nerve transmitters. It's transmitting action potential from one neuron to the next, but it's doing it via chemicals. And these chemicals are known as neurotransmitters. Okay, so the neurotransmitters are the ones that initiate the action potential in all subsequent neurons. All right. You can see here a close-up, another close-up of that. Neurotransmitters in the synaptic vesicles. Okay, they release their content into the synaptic cleft, and then they will bind to the receptor of the postsynaptic neuron and then stimulate an action potential. This slide is showing you here that a lot of drugs, whether they're drugs that affect, whether they are prescribed drugs or whether they are drugs that are being abused, whether they're illegal drugs or legal drugs, they act via the nervous system. And they have different modes of acting. So for example, we have here a drug that will stimulate the neurotransmitter to be released. So you have neurotransmitters being released very early in the process. We have drugs that will do that. We have drugs that could will cause the neurotransmitter to be released, but it could also prevent it from binding to the postsynaptic neuron. Okay, so these drugs can have many different mechanisms of action, but we're not going to focus on drug actions now. We'll look at it later this week, but I wanted to point out that these drugs, a lot of drugs, whether they are legal drugs or illegal drugs, whether they're prescribed or however you may get them, they act via the nervous system by affecting what happens at the synaptic cleft. Okay, so this is very important. Okay, Let me see how much time do we have. Oh, we have time. Oh, lots of time. Okay. Let me see if we have any questions. I guess I'll ask. Any questions so far? Okay, I'm thinking we have no questions. I will move on. Here's another slide that I would like, a video that I posted that I would like you to look at. Let me get up here. The chemical synapse. So pay attention to that, because this will show you what happens when an action potential travels all the way down to the end of the axon and gets to the presynaptic terminal, and then how it causes the release of these neurotransmitters, how neurotransmitters are released and stimulate the action potential in the adjacent neuron. Okay. All right, so now let's just move to the actual structure of the nervous system. So he said the nervous system has two sections: the central nervous system and the peripheral nervous system. Both of these components of the nervous system are made up, I should say, of what is known as gray matter and white matter. So in the brain, for example, you will see gray matter and white matter. In the spinal cord, you will see gray matter and white matter. So let's talk about what is that. What is gray matter and what is white matter? Gray matter is really just a cluster of cell bodies. That's, if you call, let's go back to a figure of the neuron. Let's go back here. Okay, so this is a neuron and this is a cell body, okay? So this is a cell body. So gray matter is clusters of these cell bodies. And they appear darker if you stain it and you look at it under a microscope, it looks darker. So it's a cluster of cell bodies that make up this gray matter. Okay, let's go back to my slide here. Okay, so gray matter made up of cell bodies and short non-myelinated fibers. Because remember we said that the axons contain myelin sheets, but there's some portions of axons or some axons that actually don't have myelin. And so short non-myelinated fibers also make up part of the gray matter. White matter, on the other hand, are made up of myelinated axons, so axons that contain myelin, and they form tracks. They form these tracks in the nervous system. So we'll focus now on what is known as on just the central nervous system, and on Wednesday we hope to get to the peripheral. We probably will get to the peripheral nervous system. So looking at the central nervous system, we said that the central nervous system is made up of the brain and spinal cord. The brain and spinal cord. Okay. Let's see here. Okay. So this is a brain. Okay. The central nervous system made up of the brain and spinal cord. So let's look at the brain. The brain actually contains 4 chambers. 4 chambers are in the brain that produce cerebrospinal fluid. Cerebrospinal fluid is produced in these 4 chambers known as ventricles. So in the brain you have 4 ventricles. It shows you here 2 lateral ventricles, a 3rd ventricle, and then what is known as a 4th ventricle. In these ventricles, cerebrospinal fluid is produced and stored. This if you have any excess cerebral spinal fluid is drained into the cardiovascular system. So under normal circumstances, excess fluid is drained into the cardiovascular system, but sometimes that doesn't always work. And you can have what is known as a condition known as hydrocephalus, which causes a buildup of the cerebral spinal fluid in the brain and can cause brain swelling. So sometimes in premature babies, you will see this. You will see some premature babies, you'll find they get a condition known as hydrocephalus, that's extra water on the brain, and it causes their heads to look very, very enlarged. And to correct that, what is done is that a surgeon will enter the brain and put a shunt in there, a tube really, into the brain and connect it to the cardiovascular system to allow drainage to occur. Okay, so that's under abnormal conditions. But under normal conditions, any excess cerebral spinal fluid that is produced is eventually drained into a cardiovascular system. So our central nervous system made up of the brain, so that's our brain there. And let's see. Then we have our spinal cord, okay? So the spinal cord, this is showing a cross-section of the spinal cord. The function of the spinal cord is communication. The function of the spinal cord is to allow for communication between the brain and the body. And you'll see in lab today, I asked the TA to show you a real brain, a preserved brain that has the spinal cord still attached to it. Okay, so the spinal cord allows for communication between the brain and the body. It functions as what is known as a reflex center also. So when you have reflex responses, it's because that information got to the spinal cord first and allows for an action to occur. And it may later to get to the brain, doesn't always get to the brain. So the spinal cord is also known as our reflex center. And this just shows you here, let's see. Well, let's look at this, okay? So this is a cross-section of the spinal cord here. Let's look at that. So imagine the cord, you took the spinal cord and you cut a slice of it out, and it's showing you that here. So here we have a gray matter, looks like a little butterfly. So that's our gray matter. And surrounding it, we have white matter, okay? And this is showing you here some of the nerves projecting off of the spinal cord. You see those nerves projecting off the spinal cord. And this is the spinal cord inside of the vertebral column. So that's the bone, the bones of the vertebra. We looked at that last couple weeks ago. And so you see the spinal cord is right in there. Okay. This is showing you the base of the skull. And that hole here, known as the foramen magnum, that is the hole through which the spinal cord enters and connects to the brain. Okay, so I want to point out, I wanted to show you this, the connection between the brain and spinal cord. So the brain is housed in the skull, and the spinal cord connects to the brain, and it enters the skull via this foramen magnum. Here's a slide that shows you that connection. So here's the brain, and this is the spinal cord attached there. Another view of it here. Brain, spinal cord attached, and you can see the nerves projecting off the cord. Here's another cross-section there. That's the spinal cord. This is white matter, and this is gray matter. Okay? That's the spinal cord inside the vertebra. That's the bones here. That's the bone, the vertebral bone, and that's the spinal cord in there. Okay? Again, more spinal cord. You can see nerves projecting off the cord. Right? I think, let's see, should we end there? Yeah. So we'll end here today. In lab, you would look at the brain. You will learn about the various segments of the brain. And we'll continue on Wednesday. Okay? Continue on Wednesday. Oh, does the use of drugs mainly prescribed or legal damage your nervous system every time you use them? Or how or why is this not beneficial in the case of headache? Using, what would you suggest, using Tylenol or not? There's no damage to the nervous system, Gadiel, if you use the if you use the drugs as prescribed. Now, there are drugs, if you use the drugs as prescribed, there should be no damage to the nervous system. But if you don't, you can eventually, over time, there are some drugs that will damage your neurons. And yes, it can lead to damage, long-term damage of some drugs if abused. In the case of a headache, I would recommend using ibuprofen, not Tylenol, because Tylenol contains, the active ingredient is called acetaminophen. And although it relieves pain, it has an effect on the liver. It has a much more damaging effect on the liver. So personally, I stay away from Tylenol because of the acetaminophen, the active ingredient. I don't use it for that reason. But it does not damage the nervous system. Tylenol does not damage your nervous system. Okay, it does other kinds of damage. So I hope I answered your question. Okay, I think that's it for today. I will see you on Wednesday and we'll go from there. Okay?